What is a literature review? To understand that, we need to return to a point that we've made earlier. Science is not a collection of facts. Rather, it's a process. It's a method for building better, more useful, more complete understandings of the world. Or, as Terry Pratchett says, science is not about building a body of known facts. It is a method for asking awkward questions and subjecting them to a reality check, thus avoiding the human tendency to believe whatever makes us feel good. It's a dynamic process filled with uncertainty and debate and controversy and struggle. It is also a human endeavor fraught with all the drama our species takes with it everywhere it goes. It is in what science doesn't know that the world becomes an enchanted place filled with mystery and wonder. Unfortunately, most of an undergraduate program focuses on learning facts. Most of the dynamics of the scientific process are ignored entirely. Today, then, I want to begin a little bit differently. I'd like to take us back through time to the year 1943. I'd like you to imagine the world without computers, cell phones, or YouTube. What's a major thing going on in the world in 1943? World War II, of course. The world was in dark, perilous times. Rather than tracking the latest news about World War II on Twitter, you'd be getting updates from the war from things like newspapers, newsreels, and probably radio broadcasts as well. But of course, there were some other nasty things going on within the United States as well. Although World War II forced open the door a tiny crack for women in the workplace, mostly women lived as second-class citizens. We also shouldn't forget that xenophobia in the United States led to the internment of Japanese Americans. Nor, of course, should we forget the gross racial inequalities that were part of the daily lived experience of all people of color. I'm not asking, then, that we return to 1943 to romanticize the past. I'm not asking us to step back into those spaces of social injustice. Rather, I'm bringing us back to 1943 because we didn't know a lot of things about the world then that we take for granted now. I want you to put yourself back into that era then in order to imagine what it was like not knowing something that you know to be true today. As an example, I'd like you to forget everything you know today about genetics and molecular biology. By 1943, we were pretty familiar with Mendelian genetics. We knew about chromosomes, we knew that hereditary traits were carried on them, and we knew that chromosomes contained both protein and DNA. We even talked a lot about genes. What we didn't know was what genes were. We had no idea what material substance carried genetic material. We were keen, however, to find out. Enter Avery, MacLeod, and McCarty, three researchers working at the Rockefeller Institute for Medical Research in New York. Avery, MacLeod, and McCarty knew a curious fact about pneumococcus. They knew that there were two varieties. R strains have a rough appearance because they lack a polysaccharide coat. S strains, by contrast, have a polysaccharide coat that gives them a smooth appearance. But that's not all that makes R and S strains different. R strains are harmless. You inject them into a mouse, and the mouse will live. Smooth strains, by contrast, are virulent. You inject S strains into mice, the mice will die. Now, heat-treated S strains are no longer virulent. You take the S strain, you heat it up to a high temperature, you then inject that into the mouse, and the mouse is just fine. So far, nothing about this story should be terribly surprising or strange. But here's where things get peculiar. If you take the rough strain and the heat-killed smooth strain and mix them together and inject that into the mouse dies. Not only that, but the pneumococcus that you retrieve from the blood is the S strain. What is it about the S strains that transforms harmless R strains to killer S strains? What is the transformative material? That's the great unknown for Avery, MacLeod, and McCarty in 1943. What is the transformative material? Importantly, Avery, MacLeod, and McCarty weren't the ones who first discovered this phenomenon. That honor belongs to Griffith, who published this weird result in 1928. The scientific community understandably met Griffith with a bit of skepticism. The simplest explanation for his strange result was that he just screwed up. And so a lot of researchers rushed in to try and replicate his findings. And his findings were verified. Neufeld and Leventhal in 1928 published a verification of his findings, as did Dawson in 1930 and Bowron in 1932. So it seems that Griffith was onto something. But just verifying the results didn't really explain what the transformative material was. To take things a step closer, in the early 1930s, several researchers found out that you don't need mice to get this transformation to happen at all. Dawson and Sia in 1931 found you could just mix heat-killed S strains with R strains and grow them up on plates, and you get S strain bacteria. Alloway took it a step further in 1932 and found that if you just took a cell-free extract, in other words, if you just lysed the cells and spun down the debris, the supernatant, what was left over, that could perform the transformation as well. So that didn't tell us what the transformative substance was, but it did tell us at least that it wasn't something in the cell walls. Now, it was always possible at this point that this is something just unique to pneumococcus, something strange that it does. And if that were the case, well, it would still be interesting because, of course, pneumonia is a dangerous disease, and knowing whatever we can about it could help in its treatment. But what if this is something that goes beyond pneumococcus? Then it speaks to something really important about the material substance of life itself. Well, in the mid-1930s, a number of researchers found out that this isn't restricted to pneumococcus at all. Berry and Dedrick in 1936 found out that rabbit viruses can also undergo transformations. Berry in 1937 found out that you can do that also in vitro. You don't need the rabbits to do that. And these findings were verified by lots of other researchers. And so this is the intellectual landscape that Avery, MacLeod, and McCarty stepped into in 1943. All of these were things that were known, but they still didn't understand what the transformative material was. And that was the big question that they wanted to answer. So, what have we done here? Obviously, I've explained the state of the art back in 1943, the cutting edge of what was understood about this strange phenomenon. But I've done something else as well. If we look at everything on the left, you see that we have a summary explanation of the state of the art, some model of the way the world works, some phenomena, in this case, how pneumococcus undergo transformations and how other organisms do as well. Here in the right, we've highlighted a gap in our understanding. Here are all the things that we know about this strange transformative process. Here's something specific that we still don't understand, a gap in our understanding. And so take these things together, and we have a literature review. This is what every single literature review does, and literature reviews are vitally important to the scientific process. They explain some model of the way the world works, and they highlight some specific gap in our understanding. That's their purpose, and that's what you'll find at the beginning of every journal article. It may be called an introduction there, it may be called the background, but at its heart, it's a literature review. So literature reviews serve two functions. They provide a summary of the latest scientific understanding of some model of the world, and they highlight a problem with that model. Providing a summary is easy for most students. You're used to writing research report papers where all you have to do is provide a summary understanding. The real trick with a literature review is highlighting a problem with what we know. And one of the biggest pitfalls is that that unknown has to be highly specific. It has to be so specific that it's something that could be addressed in a single research study. One of the big problems that students have when they get to this part of the literature review is that they're not specific enough. Time and time again, as you work on your literature review for your capstone project in VIBS 310, we will be asking you to be more specific. That's part of the process. It takes a while to winnow that down to something razor sharp in focus. And that's okay. Just remember that you're looking for something that is highly specific and something that can be addressed in a single research study. So, what's the rest of the Avery, McLeod, and McCarty story? Well, they clearly had an interesting model that they could work with, and they identified a very important gap in our understanding about that phenomenon. What was the transformative material? And so they took the cell-free extract and they treated it with proteinases, and they asked themselves, are we still able to get transformation? And the answer to that was yes. So what can they conclude from that? Well, proteinases break down protein, and so this tells us that protein can't be the transformative substance because we've broken those down. They next treated the cell-free extract with enzymes that break down carbohydrates and asked themselves again, can we get a transformation? And the answer was yes. And so clearly carbohydrates weren't part of the transformative material. Next, they treated with ribonucleases, which of course break down RNA, and still they saw transformation. And so the transformative substance couldn't be RNA. Finally, they treated with DNases, enzymes that break down DNA. And it was only at that point they stopped getting transformations. And so they concluded from that the transformative material was most likely DNA. Their findings were still subject to some skepticism because it was always possible with this methodology that the enzymes just didn't break down everything to completion. But the most likely explanation was that it was DNA. It wasn't until 1953 with Hershey and Chase and their experiments with radionucleotides that we were able to finally conclusively say that DNA was the genetic substance. But this was the landmark study that moved the scientific community from assuming that protein was the most likely genetic material to giving serious consideration to the fact that it could be DNA. And of course, we know today that DNA is the transformative substance. We know that genes are composed of DNA. But in 1943, that was far from certain. In fact, most scholars believed it was probably protein. And so that's why I always try to emphasize that science is not facts. If we just looked at the facts available to us in 1943, we'd say protein must be the genetic material. But it wasn't. And we know that it wasn't because science is a process. It's a process to constantly return to the facts and check our assumptions and put them under scientific scrutiny. Literature reviews are such an important part of this process because they highlight those areas where our models break down. They highlight those gaps in our understanding. They're absolutely critical because without gaps in our understanding, there would be no need to do scientific studies